A living human brain runs on about 20 watts of continuous power. That is roughly the draw of a small household light bulb. In that same window of time, a modern supercomputer built to model complex systems can draw tens of megawatts, most of it dedicated not to calculation itself, but to cooling the machine down so its processors do not damage themselves. The difference between those two numbers is enormous, somewhere on the order of 1,000,000 to 1. It is tempting to read that gap as proof of something dramatic, and plenty of popular science writing does exactly that. The more careful version of the claim is narrower. It is not that a computer of any kind literally cannot replicate what a brain does. It is that no one has yet found a way to replicate it using anything close to the same energy budget. And the closer researchers look at why, the more that gap starts to look like a genuine clue about the physical strategy the brain is using. Rather than a rounding error we will eventually engineer away. For decades, the working assumption in computational neuroscience was straightforward. Build bigger machines, add more processors, increase memory bandwidth, and the gap between simulation and biology would gradually close. That assumption has not been vindicated. Efforts to model the brain in ever finer detail have made real, measurable progress. But each increase in biological accuracy has come with a computational cost that grows far faster than the biological detail being captured. This is not simply a story about needing faster chips. It increasingly looks like a mismatch between two different physical strategies for handling information. One that classical silicon was never built to imitate efficiently. To understand why, it helps to start with scale, because most descriptions of the brain understate it. The human brain contains on the order of 86 billion neurons, a number large enough that people tend to stop there and assume they have grasped the complexity involved. They have not. The neurons themselves are the easy part to count. The real complexity sits in how they are wired together. Each neuron connects to thousands of others through junctions called synapses. And when researchers try to estimate the total number of these connection points across the whole brain, the figure lands somewhere around 100 trillion. For comparison, the Milky Way galaxy is estimated to hold somewhere between 100 billion and 400 billion stars. So the number of synaptic connections inside a single human skull is larger, by a wide margin, than the number of stars in our entire galaxy. That network is also not static. Synaptic strengths are constantly shifting in response to chemical and electrical signaling, on timescales ranging from milliseconds to years. A snapshot of brain connectivity, however detailed, misses the fact that the underlying structure is always in motion. Simulating a system like that using hardware that processes information in a fundamentally sequential, step-by-step fashion is a genuinely hard engineering problem. Independent of any claims about quantum mechanics, you are trying to represent a continuously evolving, densely interconnected three-dimensional system using machines that are built around a much flatter and more linear way of moving data around. The most direct historical attempt to tackle this problem head-on with classical hardware is the Blue Brain project. Launched in 2005 at the École Polytechnique Fédérale de Lausanne, the goal was ambitious even by the standards of computational neuroscience: to build a biologically detailed digital reconstruction of cortical tissue, starting with a small column from a rat brain containing roughly 30,000 neurons, reproducing the electrical and chemical behavior of that relatively small piece of tissue, tracking ion channel states, synaptic strengths, and firing patterns across all 30,000 cells at once, required some of the most powerful supercomputing resources available at the time. The project made genuine scientific contributions, refining models of cortical microcircuitry that are still used today. It also made painfully clear how steep the scaling problem is. A human brain contains roughly 3 million times as many neurons as that single cortical column, and projections of what it would take to simulate a full human brain at comparable biological detail quickly moved into territory that no currently conceivable computing facility could support. Not because the idea is impossible in principle, but because the resource requirements grow so much faster than the biological detail you are trying to capture. That mismatch became vivid in a widely cited 2013 demonstration. A team of researchers used the K computer, at the time one of the most powerful supercomputers in the world, together with a supercomputer at the Jülich Research Center in Germany, to simulate roughly 1 second of activity in a network modeled on a simplified version of cortical neurons, representing about 1% of the human brain's neuronal population, using relatively basic point neuron models rather than full biophysical detail. That simulation took around 40 minutes of processing time to complete. It is worth being precise about what that number does and does not show. It does not mean a supercomputer needs 40 minutes to think what a brain thinks in 1 second. Because the model being run was a drastically simplified approximation, not a full reconstruction of biological neurons, complete with their ion channel dynamics and dendritic geometry. What it does show is that even a stripped-down approximation of a small slice of brain activity, run on some of the most capable classical hardware that existed at the time, was already many orders of magnitude slower than the biological system it was loosely modeled on. Progress since then has been real. In more recent work using Japan's Fugaku supercomputer, successor to the K computer, researchers have simulated mouse cortical networks with close to 10 million neurons and tens of billions of synapses, and have pushed performance much closer toward real time for certain simplified models. These are genuine achievements, made possible by continued gains in processing power, more efficient simulation algorithms, and a willingness to simplify the underlying biological model in ways that trade some fidelity for tractability. Even so, the mouse cortex represents a small fraction of the complexity found in a human brain, and the fundamental architectural mismatch between a linear, sequentially organized processor and a densely dynamically interconnected biological network has not gone away. Faster classical chips have narrowed the gap in some respects. They have not closed it, and there are good theoretical reasons to think that continuing to simply scale up existing classical architectures will not close it either. This brings us back to the energy question, because it is not just a curiosity about electricity bills. It points at something built into the physics of how classical computers make decisions. Inside any conventional processor, computation happens by switching transistors between states representing 0 and 1. Every time a transistor is switched, and especially every time information is discarded rather than preserved, there is a minimum thermodynamic cost that has to be paid, and that cost is released as heat. This relationship was described by physicist Rolf Landauer in 1961 in what is now known as Landauer's principle. The principle states that erasing a single bit of information requires a minimum amount of energy dissipation, an idea that connects computation directly to the second law of thermodynamics. Modern transistors operate far above this theoretical minimum in practice, but the principle establishes that heat generation is not simply an engineering inconvenience to be optimized away. It is a fundamental consequence of doing bit erasing computation in a classical system. The brain does not appear to generate anywhere near the amount of waste heat that a system performing a comparable volume of information processing would generate if it worked the way a classical computer works. This does not, by itself, prove anything about quantum mechanics being involved. Biological neurons are simply a very different kind of information processing substrate from silicon transistors. optimized by evolution over hundreds of millions of years for energy efficiency in a way that engineered systems, built under entirely different constraints and design goals, generally are not. Much of the difference can plausibly be explained by biological neurons being highly specialized, highly parallel and analog rather than binary, using continuously graded electrical and chemical signals instead of discrete digital switching. That is already enough to explain a great deal of the efficiency gap without invoking anything exotic. Still, some researchers have pushed the question further, asking whether at least part of the brain's efficiency might come from operating closer to the kind of reversible, low dissipation regime that quantum mechanical systems can, in principle, approach. A computation that discards no information and generates no unwanted heat is a theoretical possibility within quantum mechanics, in a way that it generally is not for classical, irreversible digital logic. Whether biological neurons actually exploit anything like this is a genuinely open and contested question, not a settled one. And it leads into a much broader and more controversial area of research generally referred to as quantum biology. For a long time, the idea that the brain might rely on quantum mechanical effects in any functionally important way was treated with deep skepticism inside physics and neuroscience alike. And for a good reason. Quantum superposition and entanglement are famously fragile. To preserve them in a laboratory setting, physicists typically need extreme isolation, temperatures close to absolute zero, and careful shielding from vibration and electromagnetic noise. The brain is warm, wet, chemically busy, and constantly jostled by thermal motion. Conditions that seem about as hostile to delicate quantum states as one could design on purpose. In 2000, physicist Max Tegmark published an influential calculation, estimating that any quantum superposition involving the kind of large biological structures proposed by quantum consciousness theories would meaning lose its quantum character through interaction with its environment. On timescales of roughly 10 to the negative 13 to 10 to the negative 20 seconds. That is, many orders of magnitude faster than the millisecond timescale on which neurons actually fire and communicate. On the strength of that calculation, quantum explanations for large-scale brain function were, for years, considered essentially ruled out by most physicists working in the field. The most well-known specific proposal in this space came from physicist Roger Penrose and anesthesiologist Stuart Hameroff, who argued in the 1990s for a theory they called orchestrated objective reduction. Their proposal centers on microtubules, hollow cylindrical protein structures that form part of the internal skeleton of every cell, including neurons. Penrose and Hameroff suggested that the geometric arrangement of protein subunits inside microtubules might partially shield delicate quantum states from the surrounding thermal chaos of the cell, long enough for those states to play some functional role in cognition, and tied this specifically to a particular interpretation of quantum measurement that Penrose had proposed on independent grounds related to quantum gravity. It is important to be clear about where this theory currently stands scientifically. It remains a minority position, viewed with considerable skepticism by most physicists and neuroscientists, and Tegmark's decoherence argument, along with subsequent critiques, has never been fully answered by proponents of the theory in a way that has shifted mainstream scientific consensus. Some experimental findings, published in the years since, have complicated the picture without settling it. Researchers studying the vibrational and electronic properties of microtubules and related protein structures have reported observations of coherent energy transfer and long-lived vibrational states that are harder to explain with purely classical models than early skeptics assumed, and some laboratory studies have reported that certain general anesthetics, whose mechanism of action remains only partly understood even today, do interact measurably with microtubule structures. None of this amounts to confirmation of orchestrated objective reduction as originally proposed, and serious critics continue to argue that the relevant timescales, the warm and noisy cellular environment, and the sheer scale mismatch between microscopic quantum coherence and macroscopic cognitive processes remain largely unresolved problems for the theory. The honest summary is that quantum effects in biological systems generally, at scales far smaller and simpler than an entire thought or memory, are increasingly well documented and no longer considered fringe. Whether those effects scale up to anything resembling a mechanism for consciousness or high-level cognition is a separate and much less settled question, and treating it as already answered in either direction goes beyond what the current evidence supports. Where the evidence is considerably stronger is in a handful of much narrower biological phenomena that involve quantum effects operating over very short timescales and very small physical distances, well short of anything resembling full-scale cognition. Photosynthesis is the clearest example. Studies of light harvesting complexes in certain plants and photosynthetic bacteria have found evidence that energy absorbed from sunlight can move toward the reaction center through a process that shows signatures of quantum coherence, briefly sampling multiple possible pathways at once before settling into an efficient route, rather than simply hopping randomly from one molecule to the next, the way a purely classical description would predict. A second, reasonably well-supported example involves the magnetic sense some migratory birds appear to use for navigation, where a protein called cryptogram, found in the retina, may support a radical pair mechanism sensitive to the direction of the Earth's weak magnetic field through quantum spin dynamics. A third, more speculative example involves the sense of smell, where some researchers have proposed that the vibrational frequencies of odorant molecules, sensed through a form of quantum tunneling of electrons, might contribute to how the nose distinguishes between molecules with similar shapes but different vibrational signatures. This last idea remains considerably more contested than the first two. Taken together, these cases establish that biology is not restricted to purely classical chemistry at the molecular scale, which is itself a meaningful and relatively recent shift in how quantum biology is viewed. They do not, by themselves, establish that the brain as a whole computes using large-scale quantum coherence, and the distance between quantum effects assisting a single well-defined molecular process and quantum effects underlying entire thoughts or the experience of consciousness is considerable. Setting aside the unresolved question of whether the brain itself computes quantum mechanically, there is a separate and much better established reason quantum computers are relevant to brain research, and it has to do with what quantum computers are good at simulating, regardless of whether the thing being simulated is itself quantum mechanical in any deep functional sense. A classical bit is always definitely 0 or definitely 1. A quantum bit, or qubit, can exist in a superposition of both states at once, and when multiple qubits are linked together through entanglement, the state of one becomes correlated with the state of another in a way that has no classical analog. Measuring one instantly determines information about the other, regardless of distance. This is not simply a faster way of trying many answers one after another. It is a different mathematical structure for exploring a problem, in which the probability amplitudes associated with wrong answers can interfere destructively and cancel out, while the amplitudes associated with correct answers reinforce each other constructively. This matters enormously for a specific and very well-defined class of scientific problems, namely simulating other quantum systems. Any physical system, including individual molecules, proteins, and the electrons and atomic nuclei that make them up, can, in principle, be described by something physicists call a Hamiltonian, essentially a mathematical description of the system's energy and how it evolves over time. For simple systems, a classical computer can solve the relevant equations directly. As a quantum system grows larger, the number of variables needed to describe it on a classical computer grows exponentially, because you have to track the amplitude associated with every possible combination of quantum states the system could be in. For a system of only a few hundred interacting quantum particles, the amount of classical memory required to represent that state exceeds anything that could ever physically be built, using any conceivable amount of material in the observable universe. This is not a matter of needing faster processors. It is a mathematical wall. Physicist Richard Feynman identified this problem clearly in the early 1980s and proposed what now seems like an obvious solution in hindsight. If nature itself is fundamentally quantum mechanical, then the most natural way to simulate a quantum system is with another quantum system, one engineered so that its own physical dynamics mirror the dynamics of the thing being studied, rather than trying to calculate the consequences of those dynamics step by step on a classical machine. Instead of computing what a molecule will do, you build a quantum processor whose qubits are made to interact according to the same mathematical rules that govern the molecule's electrons, and then you simply let the processor evolve and observe what happens. The simulation and the phenomenon being simulated share the same underlying physical language. For roughly 4 decades, this remained a compelling theoretical idea without the hardware to realize it at any meaningful scale. Quantum bits are notoriously fragile, and even small amounts of noise from the environment tend to introduce errors faster than useful computations can be completed. A problem that historically got worse rather than better as systems were scaled up, since adding more physical qubits to correct errors tended to introduce new sources of error of its own. That specific problem of error correction becoming harder rather than easier as systems scale saw a genuine and well-documented breakthrough in December of 2024, when Google announced results from its Willow quantum processor. The core achievement was demonstrating that when physical qubits are grouped into larger logical qubits, error rates can decrease exponentially as the size of each logical qubit group increases, rather than increasing, which is what had generally been observed in earlier generations of quantum hardware. This is often described, reasonably accurately, as crossing a long-sought threshold in quantum error correction theory, the point at which scaling a system up makes it more reliable rather than less. It is a genuinely significant engineering result, and it addresses one of the most fundamental theoretical obstacles to building large, useful quantum computers. It is worth being precise about what it does not mean. It does not mean quantum computers are now capable of simulating anything resembling a full human brain, and claims suggesting real-time, full biological simulation of neural tissue is close at hand, substantially overstate where the technology currently stands. Willow and machines like it operate with a relatively modest number of logical qubits, sufficient to demonstrate the underlying error correction principle and to tackle narrowly defined problems in quantum chemistry, but nowhere close to the scale that would be required to simulate even a small, biologically detailed neural circuit with full molecular fidelity. What the result does establish is that the engineering path toward larger, more stable quantum processors is no longer purely theoretical. It has moved from being an open question of principle to being, at least in part, a matter of continued incremental engineering, given where the hardware actually stands. The more grounded and near-term value of quantum simulation research has less to do with simulating a whole brain, and much more to do with modeling specific molecular problems that are relevant to how brains work and how brain disease develops. Protein folding is the clearest example. Proteins are built as long chains of amino acids that must fold into precise three-dimensional shapes to function correctly. And a modestly sized protein can, in principle, adopt an almost incomprehensibly large number of possible shapes, making the search for the correct, lowest energy configuration one of the most computationally demanding problems in biology. Misfolded proteins are directly implicated in neurodegenerative diseases, including the aggregation of misfolded tau and amyloid beta proteins associated with Alzheimer's disease. Classical computers approach this problem largely through brute force search combined with approximation, which can take an enormous amount of computing time even for a single protein. The introduction of AlphaFold, an artificial intelligence system developed by represented a major leap forward for predicting the folded structure of known types of proteins, achieving accuracy that rivaled experimental methods for a large share of previously unsolved structures. It is worth being clear about what kind of tool AlphaFold is. It is fundamentally a pattern recognition system, trained on large databases of previously solved protein structures, and it performs less reliably on proteins that differ substantially from anything in its training data, and it does not directly model the physical, dynamic process by which misfolding actually happens under specific chemical or genetic conditions. A quantum computer approaches the same underlying problem from a different angle, one grounded directly in physics rather than pattern matching. In nature, a protein folds by settling into the configuration that minimizes its physical energy, continuously sampling nearby shapes until it finds a stable, low-energy state. Algorithms designed for quantum hardware, most notably the variational quantum eigensolver and quantum phase estimation, are built to find the lowest energy configuration of a quantum mechanical system directly, by using the physical qubits to represent the system and letting quantum mechanics itself do part of the search. These algorithms have already been tested successfully on very small molecules, systems with only a handful of interacting electrons, such as hydrogen and lithium hydride, producing results consistent with established quantum chemistry calculations. Scaling these methods up to proteins containing thousands of atoms, the regime relevant to real neurological disease, requires quantum hardware considerably larger and more stable than what exists today. But the underlying algorithms are already validated in principle on smaller systems, which is meaningfully different from the technology being purely speculative. If and when the hardware catches up, the practical implications for drug discovery could be substantial, potentially allowing researchers to model how a candidate's drug molecule interacts with a specific misfolded protein structure before ever synthesizing the compound in a laboratory, compressing part of a process that currently takes years and enormous expense. A second area where quantum computing intersects meaningfully with brain research, though again at an earlier and more exploratory stage than some popular accounts suggest, involves brain-computer interfaces. When a person using a neural prosthetic intends to move a limb, the relevant information arrives as noisy, probabilistic patterns of electrical activity spread across many neurons, and classical decoding algorithms typically need to accumulate enough signal to resolve that ambiguity into a confident, binary interpretation before issuing a command. A process that introduces a perceptible lag between intention and action, because a qubit can natively represent a probability distribution rather than being forced immediately into a definite state. Researchers have proposed that quantum or quantum-inspired machine learning approaches might eventually process this kind of noisy, high-dimensional neural signal more efficiently than classical decoders, reducing the latency between a person's intention and a prosthetic device's response. It is important to note this remains largely a research direction and a set of early demonstrations on limited datasets, rather than a deployed technology, and more ambitious claims, sometimes made in popular discussion, about decoding complex, abstract thoughts directly rather than relatively simple motor intentions, describe a much harder and much more speculative problem that is not close to being solved with current or near-term hardware. Stepping back from quantum computing specifically, it is worth understanding what classical neuroscience has already achieved in mapping brain connectivity, because it shows both how far the field has come and why a connectivity map alone, however detailed, is not the same thing as a working simulation. The first complete map of every neuron and synaptic connection in an entire nervous system was completed for the roundworm, a tiny organism with exactly 302 neurons and a little under 9,000 synaptic connections. Producing that map required years of painstaking manual reconstruction from electron microscope images, work largely carried out in the 1980s. Even with a complete wiring diagram in hand for such a simple organism, researchers found that classical simulations built directly from the connectivity map alone did not fully reproduce the worm's observed behavior. The map told researchers which neurons were connected to which others, but not enough about the dynamic properties of each neuron and things like how quickly a particular ion channel opens and closes, or how strongly a particular synapse responds to different patterns of input. To fully predict how the living system actually behaved, the wiring diagram turned out to be necessary but not sufficient. At the much larger scale of the human brain, the Human Connectome Project, launched in 2009 with funding from the United States National Institutes of Health, set out to map large-scale connectivity between brain regions, using a technique called diffusion magnetic resonance imaging tractography, which traces the general pathways of white matter fiber bundles connecting different parts of the brain. This approach does not resolve individual neurons or synapses. It produces a coarser, region-to-region map of how different parts of the brain are connected to one another. That map has proven genuinely useful, helping researchers understand how damage to a particular brain region tends to disrupt cognition based on that region's role within the larger network, and helping identify patterns of large-scale connectivity associated with different cognitive and clinical states. It operates at a fundamentally different resolution than anything relevant to modeling individual synaptic events, and closing the gap between a macroscale connectivity map like this and a biologically faithful simulation of neural dynamics remains a substantial, largely unsolved engineering and scientific challenge, independent of whatever role quantum effects may or may not play at the molecular level. One part of brain biology that received comparatively little attention for most of the 20th century, and has been substantially revised in recent decades, is the role of glial cells, and in particular a subtype called astrocytes. For much of the history of neuroscience, glial cells were treated mainly as structural support and housekeeping cells, supplying nutrients to neurons and helping maintain the chemical environment around them, without playing an active role in signaling themselves. That picture has changed considerably. Astrocytes are now understood to actively participate in synaptic signaling, releasing their own chemical signals, sometimes called gliotransmitters, that can modulate how strongly a nearby synapse responds to incoming activity, and they propagate slow waves of calcium activity through networks of connected astrocytes that appear to be functionally relevant to brain activity on timescales different from, but overlapping with, the much faster electrical signaling of neurons. Estimates of how many synapses a single human astrocyte can be in contact with vary considerably across studies and brain regions, but figures in the range of tens of thousands to roughly 1 million are commonly cited in the scientific literature, a striking number given that astrocytes were long assumed to be largely passive. The human brain contains roughly as many glial cells as neurons overall, and a full computational model of brain activity that leaves astrocytes and other glial cells out is, by the current understanding of neuroscience, missing a meaningful part of the picture, independent of any further questions about whether quantum effects are involved anywhere in the system. Underneath all of this sits a much older and much harder question, one that no amount of connectivity, mapping, or molecular simulation has so far resolved, which is the question of consciousness itself. Philosopher David Chalmers drew a useful distinction in the 1990s between what he called the easy problems of consciousness and the hard problem. The easy problems, despite the name, are still genuinely difficult scientific and engineering challenges, but they are challenges of a familiar kind, explaining how the visual system encodes an image, how the hippocampus stores and retrieves memories, or how the prefrontal cortex weighs different options before a decision, are all problems that can, in principle, be approached with the standard tools of neuroscience. By building better models, running more precise experiments, and gradually improving our mechanistic understanding, the hard problem is different in kind. It is the question of why any of that physical activity is accompanied by subjective experience at all. Why there is something it feels like to see the color red or taste something sweet, rather than all of that processing simply happening in the dark, functionally speaking, with no accompanying experience whatsoever. No current physical theory, quantum or classical, contains a term that corresponds directly to subjective experience, and there is no scientific consensus on how, or even whether, physical theories of this kind ever could. Several competing theoretical frameworks currently attempt to address at least part of this problem. Integrated information theory, developed primarily by neuroscientist Julio, proposes that consciousness corresponds to a system's degree of integrated information, a mathematical measure, often denoted with the Greek letter phi, of how much a system's behavior depends on the whole of it acting together rather than being reducible to the sum of independently functioning parts. On this view, a system with a high value of phi has a correspondingly high degree of conscious experience, at least in principle, regardless of whether that system is biological or artificial. Global workspace theory, associated with cognitive scientists Bernard Baars and later Stanislas Dehaene, proposes instead that consciousness corresponds to information, becoming broadly available across many specialized brain systems at once. A kind of shared broadcasting mechanism that allows otherwise separate processes to draw on the same information simultaneously. Predictive processing frameworks propose that much of what the brain does, and much of what we experience as perception, reflects an ongoing process of generating predictions about incoming sensory information and continuously updating an internal model of the world based on how those predictions succeed or fail, rather than passively receiving and processing raw sensory data from the outside in. Each of these frameworks captures something real and useful about brain function. None of them has been shown to fully explain subjective experience in a way that commands broad scientific consensus. And it is worth being direct about the fact that some researchers, including Penrose and Hameroff, have proposed that quantum mechanical processes specifically are required to bridge this gap, a claim that remains speculative and is not shared by the majority of researchers working on theories of consciousness, most of whom are pursuing explanations that do not depend on any particular claim about quantum effects in neural tissue. One place where these theoretical debates intersect with a very practical and still poorly understood medical mystery is general anesthesia. Anesthetic drugs have been used to reliably and reversibly eliminate conscious experience during surgery since the 1840s, and yet the precise mechanism by which they do this is still not fully resolved at the molecular level. more than a century and a half later, what is reasonably well established is that many anesthetic agents interact with specific classes of ions, channels and receptors in neural membranes, including gamma-aminobutyric acid receptors, and that these interactions altered the excitability and synchronization of large populations of neurons in ways correlated with loss of consciousness. Proponents of orchestrated objective reduction have specifically argued that certain anesthetics act on microtubules rather than, or in addition to, these more conventional receptor targets, and have pointed to laboratory findings of anesthetic binding to microtubule structures as supporting evidence for their theory. This remains one hypothesis among several competing explanations in an area of medicine and neuroscience, where genuine uncertainty persists. And it is fair to describe anesthesia as a case where a widely used and generally very safe medical technology continues to outpace our theoretical understanding of exactly why it works, a genuinely humbling fact for a field that has otherwise made enormous strides in understanding the brain. Away from the more speculative end of this topic, there is a large and well-established body of neuroscience describing how brain chemistry shapes cognition moment to moment. And this is worth covering in some detail, because any future simulation of brain activity, quantum or otherwise, will have to account for it. The brain does not operate at a single fixed setting. Its computational properties shift continuously depending on the levels of several key neuromodulatory chemicals, including dopamine, serotonin, and acetylcholine, each released from relatively small clusters of neurons deep in the brainstem that project widely across the brain, adjusting the behavior of enormous populations of neurons at once rather than communicating through single, targeted connections the way most other neurons do. These systems influence, among other things, how strongly the brain favors exploring new possibilities versus exploiting familiar, known solutions, how quickly learning occurs, and how strongly a given piece of information captures attention. Dopamine is probably the most widely discussed of these systems, and it is also probably the most commonly misunderstood. Dopamine is often described casually as the brain's pleasure chemical, but decades of careful electrophysiological research, particularly work associated with neuroscientist Wolfram Schwartz, beginning in the 1990s, points to a more precise and considerably more interesting role, that of a reward prediction error signal. Dopamine-releasing neurons tend to fire strongly not simply when something rewarding happens, but specifically when something better than expected happens. If an animal learns to predict a reward with high confidence, and that reward then arrives exactly as expected, dopamine neurons often show relatively little response, because there is no error to correct. If a reward is completely unexpected, dopamine neurons fire strongly. If a reward is expected and then fails to arrive, dopamine signaling drops below its normal baseline level. This reward prediction error signal appears to function as a learning signal, helping the brain adjust its expectations and reinforce behaviors that led to better than expected outcomes. And it is remarkably sensitive to small differences in timing and magnitude, a sensitivity that plays an important role in how habits form, how skills are acquired, and, when the system malfunctions, how conditions like addiction can take hold. At the level of individual synapses, one of the best studied molecular mechanisms underlying learning and memory involves a specific type of receptor called the NMDA receptor, a subtype of glutamate receptor found throughout the brain. NMDA receptors function as coincidence detectors, meaning they only open, allowing calcium ions to flow into the receiving neuron, when two separate conditions are satisfied at roughly the same time. The releasing neuron must release the neurotransmitter glutamate, and the receiving neuron must already be sufficiently electrically active from other inputs to relieve a magnesium ion that normally blocks the channel. Because both conditions have to be met together, NMDA receptors are particularly good at detecting genuinely correlated activity between neurons, rather than responding to activity that happens to occur by chance. When an NMDA receptor opens, the resulting calcium influx triggers a cascade of intracellular signaling that can ultimately lead to the synthesis of new proteins at that specific synapse, physically strengthening the connection in a process closely tied to long-term memory formation. Mutations affecting NMDA receptor function are associated with several neuropsychiatric conditions, including schizophrenia and some forms of autism spectrum disorder, underscoring how much of cognition and mental health traces back to fairly precise molecular events happening at individual synapses, well below the scale at which most current brain imaging technology can directly observe. Sleep is another area where the brain's activity turns out to be far more complex and far less understood than casual intuition suggests. During sleep, the brain does not simply power down. Synaptic connections that were active during waking hours are selectively strengthened or weakened, a process thought to be important both for consolidating meaningful memories and for pruning away less useful connections, keeping the overall system from becoming saturated with every trivial detail of daily experience. Sleep, particularly slow-wave sleep, is also associated with increased activity in a system of fluid-filled channels around brain tissue, sometimes referred to as the glymphatic system, that appears to help clear metabolic waste products, including misfolded proteins such as amyloid beta, at a higher rate than during wakefulness. Chronic sleep deprivation is one of the most reliably demonstrated ways to impair cognitive performance in humans, and disrupted sleep is associated with elevated long-term risk of several neurological and psychiatric conditions, including Alzheimer's disease, a connection that has become an active area of research given the parallel role of sleep in waste clearance. The daily rhythm underlying the sleep and wake cycle is itself governed by a remarkably precise molecular clock running inside individual cells throughout the body, including specialized neurons in a brain region called the suprachiasmatic nucleus that helps coordinate timing across the whole organism. This circadian clock is built around a feedback loop involving a small set of proteins, commonly abbreviated as clock, BMAL1, per, and cry, which regulates each other's production in a cycle that takes approximately 24 hours to complete, then resets and begins again. The precision of this cycle is genuinely remarkable given how few molecular components are involved, typically drifting by only a matter of minutes across a full day, even without external time cues. A level of reliability that took human engineers many decades of refinement to achieve in mechanical clocks, built from a much larger number of far more stable components. Mutations affecting circadian clock genes are associated with a range of health problems beyond simple sleep disruption, including elevated risk of certain cancers, metabolic disorders, and psychiatric illness, underscoring how deeply the timing of basic cellular processes is woven into overall health. Well beyond the simple experience of feeling sleepy or alert, consciousness itself appears to fade during dreamless, deep sleep in a way that is scientifically interesting precisely because so much of the brain's underlying activity continues. Metabolic waste clearance continues. Memory consolidation processes continue. Large-scale oscillatory patterns of neural activity continue in modified form. And yet, by most accounts, subjective experience is largely or entirely absent during this state, in a way that it is not during ordinary wakefulness or during REM sleep, when vivid dreaming typically occurs. identifying precisely what changes in brain activity between dreamless sleep and either waking consciousness or dreaming consciousness remains one of the most direct and empirically tractable ways researchers currently have of probing the neural correlates of consciousness, since it offers a natural, repeatedly observable transition between the presence and apparent absence of subjective experience within the very same brain, without requiring any resolution of the much harder underlying philosophical question of what consciousness fundamentally is. Another area of active research concerns not just what the brain is made of, but what kind of computational strategy it is actually running, since this shapes what a faithful simulation would need to reproduce. Classical computers are typically built to solve well-specified problems and return a single correct answer. Brains evolved for a different purpose entirely, keeping an organism alive in an uncertain, constantly changing environment, which favors fast, reasonably good decisions over slow, guaranteed optimal ones. One influential framework describing how the brain might achieve this is known as predictive coding. Under this model, the visual system, and arguably much of the rest of the brain, does not operate as a simple forward pipeline where raw sensory data flows in one direction from the eyes toward higher level interpretation. Instead, higher brain areas are understood to continuously generate predictions about what incoming sensory signals should look like, based on prior experience and context, and send those predictions back down toward earlier sensory areas. When incoming sensory activity matches the prediction, relatively little additional neural signaling is generated. When it does not match, a prediction error signal is passed back upward, updating the brain's internal model. Under this view, much of ordinary perception reflects an actively constructed internal model of the world, continuously checked and corrected against incoming sensory data, rather than a passive readout of raw sensory input. A closely related and more formal proposal, often called the Bayesian brain hypothesis, suggests that the brain is, in some approximate sense, performing something like Bayesian statistical inference, combining prior expectations with new evidence to arrive at updated beliefs about the state of the world in a way that is, at least loosely, mathematically optimal given the available information. Exact Bayesian inference over the kind of high-dimensional, richly structured information the brain deals with is computationally intractable for any classical system, which means the brain must be using some efficient approximation rather than performing exact calculations. Some researchers have speculated that quantum mechanical processes, if they play any meaningful computational role in neural tissue, might provide a natural mechanism for this kind of approximate probabilistic sampling. Since quantum systems naturally sample from probability distributions defined by their underlying wave functions, it is important to be clear that this remains a speculative hypothesis rather than an established finding. There is currently no direct experimental evidence demonstrating that the brain performs anything like quantum sampling as part of Bayesian inference, and most current research on the Bayesian brain hypothesis proceeds using purely classical, statistical, and computational models, which have already proven useful for explaining a range of perceptual and decision-making phenomena, without needing to invoke quantum mechanics at all. The quantum version of this idea is best understood as an open, testable conjecture rather than a finding that current evidence supports. If it turns out that some meaningful fraction of neural computation genuinely does rely on quantum mechanical effects, even in a narrow and specific way rather than as a grand unifying theory of the whole brain, this would have a specific and important consequence for anyone trying to build an accurate computational model. A classical simulation, however detailed, fundamentally could not reproduce that particular process correctly, because classical bits cannot represent genuine quantum superposition or entanglement. It could only approximate the statistical output of that process after the fact, using empirically fitted parameters, which is a meaningfully different and less complete kind of scientific understanding than directly simulating the underlying physics. This is the core reason quantum computing research remains scientifically relevant to neuroscience, even while so much about the extent of quantum involvement in the brain remains unresolved. If quantum effects matter anywhere in neural computation, quantum simulation is likely the only tool capable of modeling that part of the system faithfully, rather than merely describing its statistical behavior from the outside. Given the current state of both quantum hardware and biological understanding, most researchers working in this space describe the realistic near-term path not as a wholesale replacement of classical computing with quantum computing, but as a hybrid approach in which classical and quantum processors handle different parts of the same overall problem, each doing what it is naturally suited for. In such an architecture, classical processors would likely continue to manage the large-scale, relatively stable structure of a simulation. Things like the overall connectivity map of a neural circuit, input and output handling, and general bookkeeping. Tasks that classical computers already handle efficiently and reliably. A quantum coprocessor, in this picture, would be reserved for the specific subproblems where quantum effects are suspected to matter most directly, such as simulating the detailed molecular dynamics of a single ion channel, a single synaptic release event, or a specific enzyme central to memory formation. Rather than attempting to simulate an entire brain's worth of neurons quantum mechanically all at once, which remains far beyond any currently conceivable hardware. Framed this way, researchers in the field tend to describe a series of more modest, more concrete milestones as realistic near and medium-term goals. Rather than jumping straight to a full brain simulation, one widely discussed near-term goal involves demonstrating clear quantum advantage, meaning a quantum computer solving a specific, well-defined problem faster or more accurately than any classical computer practically could, for a molecule small enough to still be verifiable against classical calculations, but large enough to be biologically meaningful. Something in the range of a modest-sized region of a protein rather than an entire protein. A second commonly cited milestone involves accurately simulating the full molecular dynamics of a single type of ion channel, the class of proteins responsible for generating the electrical signals neurons use to communicate. Since ion channels are both small enough to plausibly be within reach of nearer-term quantum hardware and directly relevant to numerous neurological diseases caused by ion channel mutations. A third, somewhat more ambitious milestone involves simulating the full sequence of molecular events involved in a single synaptic release event, the coordinated cascade of calcium channel activation, protein binding, and membrane fusion that occurs each time one neuron signals to another. An event that is small in physical scale but involves a complex, tightly choreographed sequence of quantum, mechanically sensitive molecular interactions. Reaching even these more modest milestones depends on continued progress along several separate but related fronts, not just raw qubit count. Coherence time, meaning how long a qubit can maintain its quantum state before environmental noise disrupts it, needs to continue improving across the different physical platforms currently being explored, including superconducting circuits, which are relatively fast but require cooling to near absolute zero, trapped ion systems, which tend to have longer coherence times but operate more slowly, and photonic systems, which are naturally well suited to communication between quantum processors but more difficult to control with high precision. Software and programming tools also matter as much as the hardware itself. translating a well-defined scientific question into an efficient, low-error sequence of quantum logic operations is a non-trivial engineering problem in its own right, and quantum programming frameworks such as Qiskit, Cirq, and Penny Lane have been developed specifically to make this translation more tractable for researchers who are experts in biology or chemistry, but not necessarily in the underlying quantum hardware. Finally, there is the question of validation. For small systems, quantum simulation results can still be checked directly against classical calculations, providing a useful sanity check as the field develops confidence in the approach. For the larger systems, where quantum simulation would eventually provide genuinely new capability, beyond what classical computers can practically verify, researchers will need other ways of confirming that a quantum simulation is behaving correctly, such as comparing its predictions against carefully designed laboratory experiments on real biological tissue, or using known mathematical properties that a correctly functioning quantum system should preserve as an internal consistency. Check. The level of institutional investment in this general area gives some sense of how seriously parts of the scientific and technology communities are taking it, even while acknowledging how much uncertainty remains. Public funding agencies, including the United States Department of Energy, have supported quantum simulation research programs with direct relevance to biological and chemical systems, and the European Union's quantum flagship program has included the simulation of complex quantum systems among its priority research areas. Among private technology companies, Google, IBM, and Microsoft have each publicly described quantum simulation of molecular and chemical systems as a significant part of their long-term roadmaps, alongside a growing number of smaller companies focused specifically on quantum computing hardware and software. Part of the reason for this level of interest is straightforwardly commercial rather than purely scientific. Drug discovery is an industry in which even modest improvements in the speed or accuracy of molecular simulation could translate into very large reductions in cost and development time. And neurological and psychiatric conditions collectively affect a very large share of the world's population, representing a correspondingly large area of unmet medical need. It is worth noting, in the interest of giving a balanced picture, that commercial incentives structured around relatively short development and funding cycles do not always align neatly with the kind of open-ended, foundational research needed to resolve deeper questions about consciousness or the extent of quantum involvement in cognition, questions that may take considerably longer to answer, if they can be answered with current scientific methods at all. There is also a distinction worth drawing between the kind of quantum brain simulation research described here and the artificial intelligence systems that have become much more prominent in public discussion in recent years, particularly large language models and related machine learning systems. Those systems are, broadly speaking, trained by exposure to enormous amounts of existing data, learning statistical patterns in that data well enough to generate new outputs, whether text, images, or other structured content, that are consistent with the patterns they were trained on. This approach has produced systems capable of remarkably fluent and useful outputs across a wide range of tasks. But it is fundamentally a different kind of computational strategy from what quantum simulation research is attempting. A quantum simulation of a specific piece of neural tissue, if it succeeds, would not be trained on data describing how that tissue behaves. It would be built directly from the underlying physical equations governing the relevant molecules, and its behavior would emerge from those equations being physically enacted by the quantum hardware, rather than from statistical patterns learned from prior examples. In principle, this means a sufficiently accurate physical simulation could generate genuinely novel predictions about situations that were never part of any training data. In the same way, a physical model of, say, planetary motion can predict the position of a planet at a time or under conditions that were never directly observed, simply by correctly encoding the underlying physical laws. Whether current or near-term quantum hardware is anywhere close to achieving this kind of biologically meaningful physical simulation, as opposed to demonstrating the principle on much smaller test systems, remains, again, a genuinely open engineering question rather than something already achieved, looking further ahead than current hardware realistically allows. Some researchers and ethicists have begun raising questions about what obligations, if any, might eventually apply to sufficiently detailed and accurate simulations of neural tissue, particularly if such simulations were ever advanced enough to reproduce signals or dynamics closely associated with pain or distress in biological systems. This is worth mentioning honestly as a question the field is beginning to discuss, rather than as an urgent or immediate practical concern. Given how far current quantum hardware remains from anything resembling that level of biological fidelity, it is also a question without any settled answer even in principle, since it depends on unresolved deeper questions about what, if anything, gives rise to subjective experience in a physical system in the first place. Questions that remain open regardless of whether the system in question is biological, classical, digital, or quantum mechanical. Raising the question early, well before the relevant technology exists in a form that would make it urgent, is a reasonable and responsible way for the field to begin thinking about it, rather than an indication that the question is close to having practical consequences today. It is worth spending some time on the basic physics that underlies all of this, since the difference between classical and quantum descriptions of reality is genuinely fundamental, not just a matter of computational convenience. In classical mechanics, a physical system is assumed to have a definite, well-defined state at every moment, whether or not anyone is measuring it. A billiard ball has a specific position and a specific velocity, whether or not you happen to be looking at it. And if you know those values precisely enough, along with the forces acting on it, you can, in principle, predict its future trajectory with complete confidence. Quantum mechanics replaces this picture with something considerably stranger. A quantum system is described by a mathematical object called a wave function, which encodes not a single definite state, but a set of probabilities for different possible outcomes of a measurement. Before a measurement is made, the system is often said to exist in a superposition of multiple possible states at once, and the act of measurement is what selects a process usually referred to as collapse. This is not simply a statement about the limits of human knowledge, as though the particles secretly had a definite position all along and we just did not know what it was. A series of experiments testing what are known as Bell inequalities, following theoretical work published by physicist John Bell in 1964, have shown that if quantum particles actually did have hidden, predetermined definite properties before measurement, then certain statistical correlations between measurements performed on pairs of separated particles would have to fall within specific mathematical bounds. Beginning with experiments conducted by physicist Elling Aspect in the early 1980s, and repeated with steadily increasing precision in the decades since, closing various technical loopholes along the way. These experiments have consistently found that the actual measured correlations violate those bounds, ruling out a broad and important class of hidden variable explanations. The most widely accepted interpretation of these results is that quantum indeterminacy reflects something genuinely fundamental about how nature behaves at small scales, not merely a gap in human knowledge that better measurement technology would eventually close. For quantum computing, this distinction translates into something concrete and practically useful. when qubits are placed into superposition and processed together. A quantum computer is not simply running many separate classical calculations in parallel on many separate processors. The way a large classical computing cluster might divide up a problem among thousands of individual chips, it is exploring many possible computational paths simultaneously within a single, unified physical system governed by the mathematics of quantum interference, in which the contributions from paths leading toward wrong answers tend to cancel each other out, while contributions from paths leading toward correct answers tend to reinforce one another. This is why certain classes of problems, most importantly for our purposes here, the simulation of other quantum mechanical systems, can, in principle, be solved by a quantum computer using resources that grow far more slowly than would be required on any classical machine. Attempting the same calculation through direct brute force simulation, the central engineering obstacle standing between this theoretical advantage and a practically useful machine is a phenomenon called decoherence, the process by which a quantum system's delicate superposition and entanglement gradually leak away through unavoidable interactions with its surrounding environment, causing it to behave more and more like an ordinary classical system the longer that interaction continues. In a research laboratory, decoherence is fought primarily through extreme isolation, cooling superconducting quantum processes to temperatures a small fraction of a degree above absolute zero, colder than the depths of interstellar space, and shielding them carefully from stray electromagnetic radiation and mechanical vibration. Even with all of these precautions, individual qubits in current hardware typically maintain coherence for timescales measured in microseconds, 2 milliseconds, which is why sustaining a long, complex computation reliably requires the kind of layered error correction techniques that Google's Willow chip was specifically designed to demonstrate. Biological systems, by contrast, exist in a warm, wet, chemically active environment that would seem, on the surface, to guarantee extremely rapid decoherence for any delicate quantum state. One proposed resolution to this apparent contradiction, drawn from studies of quantum effects in photosynthesis and other biological processes, is that the relevant quantum events in biology may simply not need to survive for very long, since decoherence and the biological process depending on it can both occur on comparably short femtosecond timescales, meaning the quantum effect completes its functional role before the surrounding environment has time to fully wash it out. Whether anything analogous applies at the much larger scale of whole neurons communicating with each other, as opposed to isolated molecular events lasting a tiny fraction of a second, remains, once again, an open and actively debated question rather than an established fact. One useful concept for thinking clearly about what a quantum simulation of neural tissue is actually trying to achieve is substrate independence, an idea with a long history in the theory of computation more broadly. The basic insight is that a computation is defined by its logical structure and the mathematical relationships it implements, not by the specific physical material used to carry it out. The same underlying calculation can, in principle, be performed using mechanical gears, vacuum tubes, silicon transistors, or superconducting quantum circuits. And what changes across these different implementations is speed, reliability, and cost, not the abstract computation itself. Applying this idea to quantum simulation of the brain clarifies an important point that is easy to lose track of amid discussion of exotic-sounding hardware. The goal of a quantum simulation is not to build a machine that recreates the brain's actual physical conditions, its warmth, its particular chemical composition, or its specific cellular architecture. The goal is to build a machine whose internal quantum dynamics are governed by the same underlying mathematical description, the same Hamiltonian, as the biological process being studied. A superconducting quantum processor, cooled to a small fraction of a degree above absolute zero, can, in principle, still faithfully simulate the dynamics of a warm biological molecule, provided the logical structure of the simulation correctly captures the relevant physics. Because what matters for the validity of the simulation is the underlying mathematics being reproduced, not whether the physical conditions of the hardware happen to resemble the physical conditions inside a living cell. This distinction between physical resemblance and logical correspondence also helps explain why quantum simulation research and large-scale classical connector mapping are complementary research programs rather than competitors, addressing different levels of description that will eventually need to be combined rather than choosing between them. Large-scale connectivity mapping, of the kind pursued by the Human Connectome Project and similar efforts, provides the overall architectural blueprint, showing which brain regions and, at finer resolution, which individual neurons are connected to which others, and roughly how strongly. Quantum simulation, where it eventually becomes practical at meaningful biological scale, would provide a way of faithfully executing the fine-grained molecular dynamics happening at individual synapses and within individual proteins. The level of detail at which quantum mechanical effects, if they matter functionally at all, are most likely to be relevant. Neither approach, on its own, constitutes a complete simulation of brain activity. The connectivity map without accurate dynamics is a static diagram. Accurate molecular dynamics without an overall connectivity map has no larger structure to operate within. Progress on both fronts, pursued somewhat independently by different research communities using very different tools, will eventually need to converge for either to reach its full potential. The feedback loop between a neuron's ongoing activity and its underlying molecular machinery is itself an important and often underappreciated part of this picture, and it directly connects the study of memory back to the study of gene expression and protein synthesis. When a pattern of neural activity is significant enough, meaning frequent enough, strong enough, or correlated enough with other relevant signals, it can trigger changes in which genes a neuron expresses over the following hours and days, altering the mixture of proteins present in that neuron and, in turn, altering its physical and electrical properties going forward. This means a neuron that has recently experienced a particular pattern of meaningful activity is, in a literal molecular sense, a physically different cell some hours or days later than it would otherwise have been, a process widely regarded as one of the core molecular mechanisms underlying long-term memory storage. This process is also highly selective. Synapses that carry meaningful, correlated patterns of activity tend to be marked for structural strengthening through mechanisms closely tied to the NMDA receptor coincidence detection process described earlier, while neighboring synapses carrying more random or uncorrelated background activity generally are not. The molecular events responsible for this selectivity, distinguishing a synapse experiencing a genuinely meaningful pattern of correlated activity from one experiencing background noise, involve exactly the kind of fast, precisely timed molecular interactions, protein binding, conformational changes, and ion channel gating that sit at the boundary between classical and quantum descriptions of chemistry, and that a sufficiently advanced quantum simulation would, in principle, be well positioned to model with a level of physical fidelity that current classical approximations cannot fully match. It is worth placing all of this within the broader history of scientific instruments, because that history offers a useful, more measured way of thinking about what a mature quantum simulation platform for neuroscience might eventually provide, without overstating where things currently stand. The invention of the telescope allowed astronomers to observe details of the solar system, such as the moons orbiting Jupiter and the cratered surface of the moon, that were simply invisible to unaided observation. And those new observations played an important role in overturning long-held assumptions about the structure of the cosmos. The invention of the microscope revealed that living tissue is composed of individual cells, a discovery that eventually reshaped medicine's basic understanding of disease. The development of functional magnetic resonance imaging in the late 20th century allowed researchers, for the first time, to observe patterns of brain activity associated with different cognitive tasks in living human subjects without invasive surgery, contributing to a substantial revision of earlier, more simplistic views about specific mental functions being narrowly localized to single, fixed brain regions. In each of these cases, a genuinely new instrument revealed previously invisible aspects of reality, and those revelations tended to be at least partly unpredictable in advance, which is part of why building the instrument mattered in the first place. If researchers could have fully anticipated what a new instrument would show, the instrument itself would have added comparatively little, a mature quantum simulation platform for neuroscience. If the current research program succeeds over the coming decades, would plausibly belong in this same broad category. An instrument capable of revealing molecular-level dynamics inside neural tissue that are currently invisible to every existing experimental technique, whether that is functional imaging, which operates at too coarse a spatial and temporal resolution, or electrode recordings, which can achieve high resolution but only within a very small, physically accessible region at a time. It is worth being appropriately cautious rather than triumphant about this comparison, however. The telescope and microscope were relatively simple optical devices built from well-understood physical principles, refined incrementally over generations, rather than requiring a fundamentally new branch of physics and engineering to function at all. A quantum simulator capable of meaningfully modeling biological neural tissue requires maintaining large numbers of extremely fragile quantum states in a stable, controllable, and verifiable way, a task that remains, even after the genuine and significant progress represented by results like Google's Willow chip, a substantial unsolved engineering problem rather than an incremental refinement of already mature technology. The historical analogy is useful for thinking about what kind of scientific value such an instrument could eventually provide. It should not be read as a prediction of exactly when, or with total confidence that the necessary engineering will actually be completed on any particular timeline. Taken together, the various threads covered here, the persistent energy efficiency gap between brains and classical computers, the genuine but narrowly scoped evidence for quantum effects in specific biological processes, like photosynthesis and possibly bird navigation, the unresolved and still actively contested status of more ambitious theories like orchestrated objective reduction, the real and significant but still early-stage progress in quantum hardware exemplified by Google's Willow chip, and the more concrete near-term applications in areas like protein folding and drug discovery, point toward a field that is genuinely active and genuinely promising, without yet having delivered anything close to its most ambitious, long-term goals. That is a normal, healthy state for an active area of scientific research to be in, not a sign that the underlying questions are unimportant or that the field is overhyped in every respect. It does mean that confident claims, in either direction, that the brain has been proven to be a quantum computer, or alternatively that quantum effects in biology are definitively irrelevant to cognition, go beyond what the current evidence actually supports, and a careful, accurate account of where the science currently stands has to sit somewhere between those two extremes. It is also worth addressing a reasonable question that tends to come up in discussions like this one, which is why researchers are pursuing quantum simulation at all. rather than simply continuing to improve classical computing. Architectures that are specifically designed to resemble biological neural networks more closely. This is not a hypothetical alternative. It already exists in a research area called neuromorphic computing, which builds specialized classical hardware whose physical architecture is deliberately modeled on features of biological neural circuits, including highly parallel, event-driven processing where computation happens locally and only when needed, rather than following the traditional approach of continuously cycling data back and forth between a separate processor and memory unit. Systems like Intel's Loy chips and IBM's True North architecture represent serious, ongoing engineering efforts in this direction, and they have demonstrated meaningful energy efficiency improvements over conventional processors for certain brain-inspired computing tasks, in some cases by several orders of magnitude compared to standard classical hardware running equivalent workloads. Neuromorphic computing is a genuinely valuable and actively developing research direction in its own right, worth mentioning on its own terms, rather than only in contrast to quantum approaches. It is important to be clear, however, about what neuromorphic computing can and cannot address. Neuromorphic chips remain fundamentally classical devices, built from ordinary transistors. However cleverly arranged and however specialized their architecture is for brain-like workloads, they can dramatically improve the energy efficiency and speed of simulating certain aspects of neural network behavior, particularly the kind of large-scale, statistically described spiking neural network models that are already tractable in principle on conventional computers. What they cannot do, for the same fundamental reason ordinary classical computers cannot do it, is natively represent genuine quantum superposition or entanglement. If such effects turn out to matter for any specific piece of biological neural computation, if it turns out that the relevant computation happening in real neurons is purely classical in every functionally important respect, simply implemented through biological rather than silicon hardware, then neuromorphic computing, refined over enough engineering generations, might eventually be entirely sufficient for building highly accurate, highly efficient brain simulations without any need for quantum hardware at all. If, on the other hand, some meaningful fraction of neural computation does rely on quantum mechanical effects in a functionally important way, no amount of refinement to a classical neuromorphic architecture, however brain-inspired its overall design, could close that particular gap, because the gap in that specific case would not be one of architecture or efficiency, but of the underlying physical resources available to represent the relevant states in the first place. This is precisely why the current uncertainty about how much, if any, of neural computation genuinely depends on quantum effects matters so much for deciding which long-term research investment is likely to pay off, and why serious researchers are pursuing both neuromorphic and quantum approaches simultaneously, rather than betting everything on either one. There is one further piece of the quantum computing landscape worth mentioning: quantum networking. Since a full-scale simulation of a large biological system would likely require more computational resources than any single quantum processor, however advanced, could provide on its own. Quantum networking research aims to connect multiple separate quantum processors together through quantum communication channels in a way that can, in principle, preserve entanglement and coherence across physically separate devices, allowing them to function collectively as a larger distributed quantum system rather than as isolated islands of computation. Achieving this reliably requires its own dedicated hardware, including devices called quantum repeaters that can extend entanglement across longer distances without simply measuring and destroying the fragile quantum states involved. and specialized quantum memory devices capable of holding a quantum state stable for long enough to synchronize with other parts of a distributed system. Quantum networking research is being pursued partly for entirely separate reasons, particularly applications in quantum cryptography for secure communication, which gives it a broader base of research investment than brain simulation alone would provide. A genuinely functional, large-scale quantum network, if and when it is built, would also be directly useful for distributing the kind of large-scale biological simulation being discussed here, allowing different quantum processors to each handle a local region of simulated neural tissue while remaining meaningfully connected to the rest of the system, in a rough structural echo of how the brain's own computational power emerges from a network of individually limited components working together rather than from any single, especially powerful node. Psychiatric medicine offers a particularly concrete illustration of why more precise molecular simulation, whether achieved through quantum computing, improved classical methods, or some combination of the two, could have real practical value well before anything resembling a full brain simulation becomes possible. Many of the most widely used psychiatric medications work by altering neuromodulatory signaling in some way. Antidepressants that affect serotonin availability, antipsychotic medications that target dopamine receptors, and stimulant medications that enhance both dopamine and norepinephrine signaling, among others. In practice, these drugs were developed and refined largely through a combination of informed hypothesis, animal testing, and controlled clinical trials measuring outcomes in patients, rather than through a complete, ground-up molecular understanding of exactly how a given drug alters the relevant signaling cascade at every step. This approach has produced genuinely effective treatments for many patients, but it also means that predicting in advance which specific molecular modification to an existing drug might improve its effectiveness or reduce its side effects for a particular condition remains, in significant part, a matter of trial and error, an approach that is slow, expensive, and does not always generalize well from one patient population to another. A simulation capable of accurately modeling the full molecular cascade triggered when a specific drug molecule binds to its target receptor, including every subsequent conformational change and downstream signaling step, would represent a meaningful shift toward a more physics-grounded, more predictable approach to psychiatric drug development. Though it is worth being clear that even a successful demonstration of this kind of simulation for one specific drug target would represent a significant but incremental step, not a wholesale replacement for the extensive animal and human testing that responsible drug development still requires for safety and efficacy reasons that go well beyond molecular mechanism alone. Stepping back to consider the overall shape of this research area, a fair summary would note several distinct threads that are related but should not be collapsed into a single overstated claim. First, there is a genuine and increasingly well-documented body of evidence that some specific, narrowly defined biological processes, most clearly photosynthesis, and quite possibly avian magnetic navigation, involve quantum mechanical effects operating on very short time scales and very small physical distances. Second, there is a considerably more speculative and still scientifically contested hypothesis, most closely associated with Penrose and Hameroff's orchestrated objective reduction theory, proposing that quantum effects inside neurons play a direct functional role in cognition and consciousness specifically, a hypothesis that faces serious, still largely unanswered theoretical objections regarding decoherence timescales, and that remains a minority position within both physics and neuroscience. Third, entirely independent of whether the brain itself turns out to compute quantum mechanically in any interesting sense, Quantum computers are becoming genuinely useful, well-validated tools for simulating certain classes of molecular and chemical systems relevant to neuroscience and medicine, including protein folding and drug receptor interactions, an application area with real, near to medium-term practical value that does not depend on resolving the more speculative questions about consciousness at all. Fourth, and finally, foundational questions about the physical basis of subjective experience, the hard problem of consciousness, remain genuinely unresolved by any current theoretical framework, quantum or classical, and are likely to remain at least partially open even as quantum simulation technology continues to mature and generate increasingly precise data about the physical processes occurring inside neural tissue. Keeping these four threads distinct, rather than treating progress on one as automatically validating claims about another, is essential for accurately understanding where this genuinely fascinating area of research currently stands. Looking ahead at what would actually move this field from open question to settled science, several specific lines of research stand out as the ones most likely to matter over the next decade or two. On the biology side, researchers studying quantum effects in microtubules and related cellular structures need higher resolution experimental techniques capable of directly measuring quantum coherence inside living, functioning neurons under normal physiological conditions, rather than in simplified laboratory preparations using isolated proteins removed from their native cellular environment. Techniques like ultrafast spectroscopy, which can track molecular events on femtosecond timescales, and increasingly sensitive quantum sensing methods, originally developed for entirely different applications, are gradually being adapted for exactly this purpose, and results from this work over the coming years should meaningfully narrow the range of remaining uncertainty, in one direction or the other, about how significant quantum effects actually are inside functioning neural tissue. On the hardware side, the next major benchmarks to watch involve scaling logical qubit counts well beyond current levels, while maintaining the favorable error correction behavior first demonstrated with Google's Willow chip, along with continued improvements in coherence time across competing qubit technologies, and the first genuinely convincing demonstrations of quantum advantage for molecular simulation, problems that are directly relevant to biology rather than more abstract mathematical benchmarks designed primarily to showcase raw computational power on the theoretical side, competing frameworks for consciousness, including integrated information theory, global workspace theory, and predictive processing models, are gradually being refined into more precise, more experimentally testable forms. An important development given that vague theoretical frameworks are difficult to confirm or rule out no matter how much additional data becomes available. As quantum simulation tools mature, even in their current early form, they offer a genuinely new way of generating data that can help distinguish between these competing frameworks, not by resolving the underlying philosophical hard problem through argument alone, but by providing increasingly precise physical descriptions of what changes in the brain, at the molecular level, as a person moves between different states of consciousness, from ordinary wakefulness through dreaming sleep to dreamless deep sleep and back, or through the gradual onset and later reversal of general anesthesia. None of these lines of research are likely to produce a single, dramatic, all-at-once resolution to the deepest open questions discussed here. Progress in this area, as in most areas of serious science, is more likely to arrive gradually through a steady accumulation of better measurements, more refined theories, and incrementally more capable instruments, each contributing a piece to a picture that will probably take considerably longer than a single decade to come fully into focus. What can be said with reasonable confidence today is that the underlying physics motivating this entire research program is sound. Biological molecules genuinely do operate at length scales where quantum mechanical effects are not optional. Extra details, but a fundamental part of accurately describing how they behave. A fact well established, independent of any of the more speculative claims discussed here about consciousness specifically. Classical computers genuinely do face a fundamental, not merely practical, mathematical barrier when trying to simulate sufficiently large and richly interconnected quantum systems directly. A barrier first clearly articulated by Feynman more than 4 decades ago and confirmed repeatedly since. And quantum computing hardware genuinely has made significant, well-documented engineering progress in recent years, particularly around error correction. Progress that puts the field on a considerably more solid practical footing than it occupied even a few years earlier. None of this guarantees that quantum computing will ultimately deliver a complete, biologically faithful simulation of a human brain. And researchers working seriously in this area are, for the most part, appropriately cautious about time, generally describing the goals discussed here as a multi-decade research program rather than something likely to be achieved within the next several years. It does mean the underlying scientific reasoning connecting quantum mechanics, biology, and computation is grounded in solid, well-established physics rather than speculation for its own sake, which is precisely what makes this an area worth following carefully as the research continues to develop, rather than dismissing it either as science fiction or accepting its most dramatic claims at face value, taken as a whole. The story here is less about a single dramatic breakthrough and more about several separate, genuinely difficult scientific and engineering problems, slowly converging on the same set of questions from different directions. Physicists studying error correction are making quantum hardware more stable. Biologists studying photosynthesis and bird navigation are documenting quantum effects operating in real living systems, under real environmental conditions, rather than only in carefully isolated laboratory setups. Neuroscientists studying memory, sleep, and consciousness are producing increasingly precise descriptions of exactly what needs to be explained, narrowing the target that any future simulation, quantum or otherwise, will eventually need to hit. None of these threads on its own settles the biggest open questions about whether the brain relies on quantum mechanics in any functionally important way beyond the ordinary quantum chemistry present in all molecular biology, or about what, if anything, a sufficiently detailed simulation of neural tissue would tell us about the nature of subjective experience. Together, they represent a genuinely active, genuinely evidence-driven area of science, moving forward through careful incremental progress rather than through overstated claims in either direction. If you found this walk through the physics, the biology, and the genuinely open questions worth your time, following how this research develops over the next several years is likely to be worthwhile, since each of the specific milestones described here, in error correction, in molecular simulation, and in quantum biology experiments, is concrete enough to be checked against as the results actually come in. It is also worth saying a word about how this topic tends to be covered in popular science media, since that coverage shapes public expectations in ways that are not always accurate. Headlines describing a single new result as proof that the brain is a quantum computer, or as evidence that quantum computers will soon simulate human consciousness, generally overstate what the underlying research paper actually claims, often considerably. The scientists producing this work are, in general, far more cautious in their own publications than the headlines summarizing those publications tend to be, careful to describe specific, narrowly scoped results. A particular molecule simulated, a particular error correction threshold crossed, a particular vibrational signature measured in a particular protein, rather than sweeping claims about consciousness or artificial general intelligence. A useful habit, when encountering a striking headline in this area, is to ask specifically what was actually measured or calculated, on what system, at what scale, and how that particular result connects, if at all, to the much larger and much more speculative claims often layered on top of it in secondary coverage. Applying that same standard consistently to every claim discussed throughout this account, from the energy comparison between brains and supercomputers, through the status of orchestrated objective reduction, to the current capabilities of Google's Willow chip, is the most reliable way to maintain an accurate picture of where this fast-moving and genuinely important area of research actually stands today, and where it still has a considerable distance left to travel before its biggest questions are resolved.